Welcome to Renal Grand Rounds. I think we'll get started so we can get everyone where they need to go on time. So it's my pleasure today to welcome Dr. Paolo Cravedi, who is a transplant nephrologist for Renal Grand Rounds. Dr. Cravedi currently is at Mount Sinai School of Medicine in New York. He is a transplant nephrologist, but also a basic scientist. Got his MD in near Milan, where he's from, and then went on to do his some additional research work both in Bergamo and also in Bethesda at NIH, before he went and got his PhD at Open University in London in transplantation research, and then has been in the US and been for a long time now at Mount Sinai. He's been a fantastic collaborator, not just with us here at Northwestern, but is a major collaborator of Dr. Levitsky on his CTOT liver studies. And we're going to hear about an interesting new pathway they're studying. And actually, they were just awarded a ITN grant for this, looking at the role of erythropoietin as a hormone that can modulate the immune system. And so welcome, Dr. Cravedi.
Thank you, John. And thank you, everybody, for being here, for having me today. It's a real pleasure meeting in person collaborators and friends that I meet on a weekly basis. Today we're gonna discuss about this project that started a few years ago, and about the immune modulating effects of erythropoietin. So a couple of historical notes on EPO. Was over a century ago when two French scientists reported that plasma from rabbits with anemia was able to increase the red blood cell production when it was injected into anemic animals. And was only in the mid-50s that the hormone that was responsible for this effect, erythropoietin, was isolated. Became clear that the kidney is the main source, not the only one, but the main source of erythropoietin or EPO production, and the main driver is hypoxia. So we started working on erythropoietin when we saw the results of this trial. So this is a randomized controlled study done in France, in which kidney transplant recipients were randomized to have a full or partial correction of the hematocrit. And the results were striking. So the full normalization of hematocrit was associated with a significantly higher graft survival at 2 years. And the main explanation, the hypothesis, was that the full normalization of the hematocrit was improving the oxygenation of the graft, so improving the survival. But we thought that maybe there was also something more, that EPO had some immune modulating effects. And so when I was in Italy, my initial mentor, Remuzzi, did this very interesting and elegant study. So he used this chronic kidney graft rejection model in rats. And he used proteinuria as a readout of chronic rejection. And he treated rats with EPO, and he saw that there was less proteinuria, there was less chronic rejection. And then he did this very elegant control group in which he transfused the rats with red blood cell from rats. So the full normalization of the hematocrit without EPO was not able to reduce the proteinuria. So indicating that the effects, the nephroprotective effect of EPO were independent from the erythropoietic effects. And then we looked in the literature, and others have also worked on erythropoietin, and this is a study in EAE, autoimmune encephalomyelitis in mice. And as you can see, the use of erythropoietin is associated with late onset of the disease. Here is the severity score, and with milder severity of the disease. So all these studies were done in rodents. We started wondering whether also human cells express the EPO receptor. And these are T cells, human T cells. At baseline, the expression level of the EPO receptor mRNA is very little. But if we activate the cells with anti-CD3, CD28, there's a significant upregulation of the gene and also of the protein. So our immune cells, our T cells, have the EPO receptor. Is it functional? So when we stimulated the cells and we labeled them with CFSC, we added EPO, we saw that progressively the proliferation of the cells here that you can see here marked as the dilution of the CFSC progressively declined. So declined in a dose-dependent mode. And this was true both for the naive and for the memory CD4 T cells. And this is important because the memory cells are the ones that are most important in the allograft rejection, and only calcineurin inhibitors are truly effective in inhibiting these cells. So if EPO has this inhibitory effect, we ask what are the mechanisms responsible for this. And this is a schematic that is the results of many years of studies. And what we found is that the EPO receptor has a JAK2 signaling that activates a phosphatase, a SHIP1, so removes the phosphate. And through a crosstalk mechanism, inhibits the signaling downstream IL-2. IL-2 is one of the key driver for T-cell T-cell expansion. So inhibiting IL-2 signaling is a key mechanism how EPO prevent the T-cells from expand. But then when we realized that, we also realized that IL-2 is also important for the Tregs, the cells that regulate effector T cells. And so we were worried that EPO inhibits the proliferation of effector cells, but may inhibits also the Treg. And so we repeated the same experiment. We activated effector and Treg with anti-CD3, CD28. And then we added EPO, increasing doses of EPO. As you can see here, the effector cells proliferated less and less when we gave EPO, but the Treg were totally unsensitive. So luckily, EPO was not effective on Tregs, was not inhibiting T cells, Tregs. So the first question we asked is whether the lack of inhibitory effect on Treg of EPO was due to the fact that these T cells do not express the EPO receptor. So we stained here in green for the EPO receptor on Tregs. And as you can see, the Tregs express the same level, if not higher level of the EPO receptor. So this could not be the mechanism explaining why EPO is not affecting these cells. And then we went back to the signaling downstream the IL-2 receptor. There's an alpha and a beta chain and a gamma chain. And the gamma chain is important for the STAT5 phosphorylation, which is important for the maintenance and the proliferation and well-being of the Tregs. And we realized that the phosphatases that are inhibited by EPO are already shut down, already silenced in the Treg. So EPO is inhibiting a signaling that is already silenced in the Treg. So that's why if we give EPO, these cells are not sensitive. Tregs come in different flavors. There's two main group of Tregs, the ones that are originated directly from the thymus, the thymic Tregs, the most stable ones, and the ones that are originated in the periphery. We showed that the EPO has no effect on the cells that are already Treg, but now we started asking whether EPO can affect the conversion from a naive into induced Treg. And our hypothesis, based on the fact that EPO promotes the TGF-beta production in other cells, was that EPO could act on the antigen-presenting cells, favoring the transition from a naive into a Treg. So, and this is the experiment we did. We took antigen-presenting cells, macrophages, with IL-2. This is the amount of Tregs that we can generate. The FOXP3 is a hallmark of Tregs. When we added EPO, there was a significant upregulation of the FOXP3 expression to the same level of the positive control with TGF-beta. Then we isolated these cells, the FOXP3 positive cells, and we tested their function in vitro. We did suppression assay. And these cells were as good as the positive control, as the one that were generated in the presence of TGF-beta. So EPO helps the induction of Tregs that are functional. And so this was our hypothesis that this works through the production of active TGF-beta. And we tested that. We tested the production of TGF-beta in EPO-stimulated macrophages. We saw that it went up. The TGF-beta has to be activated to be functionally active. We saw that there was an increase also of the active portion of the TGF-beta, and also of these enzymes, one of the enzymes, the UPA, that cleaves the tail of the TGF-beta, making it functionally active. And when we block the TGF-beta receptor, or this enzyme, the increase in the Tregs that you can see here was abolished. So indicating that TGF-beta APO is really inducing Tregs through this TGF-beta mediated mechanism. We mentioned before that the thymic Tregs are stable. So they are originated from the thymus, and they stay Treg for all their life. But the Tregs that are generated in the periphery are unstable, and they can convert into effector cells, like in Th17 cells and other effector cells. So we tested the hypothesis that EPO was able not just to promote the induction of Tregs, but also to increase the stability of these cells. So we used these mice that do not have an immune system, and we injected human cells. And we injected Tregs in these mice together with other effector cells that could represent a source of IL-2. And then we treated the mice with or without EPO. We injected cells that were over 95% FOXP3 positive. And after 3 days, 1/3 of the cells lost the FOXP3 expression, while the EPO administration preserved their FOXP3 expression. So indicating that EPO is also allowing these Tregs, these induced Tregs, to be more stable in the periphery, which is very important because some of these Treg, when they are injected in the context of rejection, for example, they can convert into effector cells. So since the kidney is the main source of EPO, we started wondering whether the EPO is responsible for the immune modulating effects of the kidney. So you know that HIF is the key transcription factor that senses the presence of oxygen, and the activation of HIF drives the EPO production. So at that time, we didn't have EPO knockdown animals, so we gave HIF inhibitor, acrylflavin, to mice, and we tested whether by inhibiting the production of EPO in mice, we could reduce the immune modulating effects of the kidney. So we transplanted kidneys from bulb C into black 6 mice, RAG knockout mice that don't have TMB cells. And then we injected CD4 cells that can recognize the bulb C antigen. And we saw how many of these cells became Tregs. So these are the cells that we injected. After 1 week, 3.5% of the cells became Treg. So the kidney is able to transform naive cells into Tregs, okay? So has immune modulating effects. But if we give acrylflavin, if we block the EPO production by the kidney, we prevent this effect. So this suggests that EPO is implicated in this, in the Treg formation, okay, induced by the kidney. Is this relevant? So we did kidney transplant, and there are some strain combination in mice that are spontaneously tolerated. That doesn't happen in humans, unfortunately, but happens in mice. And so these are the mice that are tolerated for over a month. If we treat the mice with acrylflavin, if we block the production of EPO, this kidney transplant gets rejected. And this is not because the acrylflavin is injuring the kidney per se, because if we use isografts, okay, acrylflavin is not doing absolutely anything. And if we give EPO to the mice that were treated with acrylflavin, we restore the EPO by giving exogenous EPO, we could restore these prolonged acceptance, okay? These are kidneys that are nephrectomized. These are mice that are nephrectomized, so it's the survival is really checked by the survival of the mice. We also checked into the kidney, and all these little spots are FOXP3 positive cells, are Tregs. And as you can see, when we block EPO, the Tregs are no longer there. And when we block endogenous EPO, when we give exogenous EPO, these Tregs appear again. And here is a quantification of the results. Spontaneous kidney transplant tolerance, unfortunately, does not occur in humans, as was just said, and as you know well. But if EPO is responsible for this phenomenon, it's possible to speculate that exogenous administration of EPO has some protolerogenic effects, regardless from the kind of transplanted organ. So we went back to the lab, and now we transplanted heart into mice. Heart transplant are rejected quickly. We gave these cells that recognize the heart antigen. And after 5 days, we measured FOXP3 expression. Now, as you see, if you transplant heart, no cell, very little cells become Treg. So the heart that is not as special as the kidney, does not convert naive cells into Tregs. But if we give exogenous EPO, a tiny fraction, but a significant fraction of these cells become Treg. Now, is this relevant? As I said, these hearts are rejected in about 1 week. But if we give EPO, if we just give 3 doses of EPO at the time of transplant, kind of as an induction, we prolong the graft survival. And this was associated with more Tregs and fewer effector T cells. When we gave EPO together with CTLA-4 Ig, this is if we give only belatacept, we have a prolongation, but the CTLA-4 Ig plus EPO further synergized to allow the survival of the grafts for over 2 months. In these experiments, we gave EPO just here at the beginning, just as an induction, just 3 doses of EPO. If we keep treating the mice with EPO for up to 60 days, most of them do not reject. Most of the mice do not reject. The heart is still beating. This is a heterotopic heart transplant, but it's still there. But if we do histology, if we sacrifice the mice at 100 days, we see that this is not true tolerance. There are infiltrates, and these are not only Tregs. There's probably an ongoing chronic rejection. But still, the mice with only EPO can survive for over 100 days. So altogether, these data provide a new interpretation of previously reported protolerogenic effects of kidney transplant compared to other organs. For example, if we look at the incidence of rejection in heart transplant recipient versus heart plus kidney transplant recipients, the incidence of rejection is much lower in recipients of kidney or combined heart and kidney transplant recipients. This is an analysis that has a lot of caveats, but is not associated with, is not due to the fact that these patients are receiving higher immune suppression, because immune suppression in the 2 cases is very similar. And in pigs, the heart is rejected in about 1 month, the lungs in about 2 months. The kidney is not rejected if we give 2 weeks of cyclosporin. Interestingly, if we give kidney plus the heart, and the heart and the kidney come from the same donor, also the heart gets tolerated. So there's something special in the kidney that protects the heart, and we think that EPO is one of these elements. So overall, the data so far indicate that EPO has a direct inhibitory effect on effector cells, prevents their proliferation, but also acts on the antigen-presenting cells, promotes the release of TGF-beta and the conversion of naive cells into Tregs. We don't know what is the most important mechanism, although, to prevent rejection. Is the direct effect on the effector, or is the promotion of the Tregs? So together with Peter Higer, we generated these mice that are conditional knockout. So they do not express the EPO receptor selectively on the T cells or on the myeloid cells. So they do not express the EPO receptor here or here, to test which one is the dominant pathway. And we transplanted, we gave a heart transplant, and here are the results. If we gave CTLA-4 Ig plus EPO, there's a prolongation of the graft survival over CTLA-4 Ig alone. But if the mice do not express the EPO receptor on the myeloid cells, the effect of EPO is prevented. So indicating that most of the protolerogenic effects are mediated through the antigen-presenting cell, through the myeloid cells. Because when we removed the EPO receptor on the T effector cells, EPO was not really able so actually, yes, we didn't see this beneficial effect. Then we did another experiment. We focused on the naive T cells. We wanted to test if the effect of EPO was really mediated by the antigen-presenting cells. And what we did, we gave transplant and then erythropoietin, and we saw an increase of the Tregs. But when the mice were not expressing the EPO receptor on the myeloid cells, not only the graft survival was no longer there, but also the increase in the Treg was no longer there. So again, indicating that the effect is mediated by the antigen-presenting cells. We also tested the transcriptional profile of these cells. We saw that when we gave EPO, the macrophages of the mice start to express a different transcriptional profile. And primarily what we saw was an upregulation of an inhibition of the pro-inflammatory cytokines in the mice treated with EPO, and a downregulation of all the pro-inflammatory cytokines, again, in the mice treated with EPO. So indicating that there's not just increased production of TGF-beta, but there's a whole transcriptional program that is proteolerogenic in mice treated with EPO. So we started wondering whether myeloid cells could be a target of EPO. If we give another costimulatory blockade, anti-CD40 ligand, the mice accept their graft for over 2 months. But if they receive CpG, which is a toll-like receptor agonist, which is similar to what would happen when we have a bacterial infection, the tolerance, the graft acceptance is broken and the mice start rejecting. So we wondered what happens if we give CpG together with erythropoietin. Can we restore graft acceptance? And this was indeed what we found. Not all the mice were able to accept the graft at 60 days, but a significant fraction of them did. And this happened only in the mice that were expressing the EPO receptor on the myeloid cells. If they don't express the EPO receptor on the myeloid cells, EPO is no longer effective. And we started reasoning that maybe this data could not simply be explained by the fact that when we give CpG, this toll-like receptor agonist, EPO is preventing the activation of these cells, but maybe EPO is acting on a deeper level. So, there is this concept of trained immunity. So when macrophages see an antigen, this induces changes in their epigenetic profile, so that when they are exposed to a similar, not necessarily the same, but to a similar antigen in the future, they can be activated much, much faster. Okay, that's a concept of trained immunity, which is a little bit different from immune memory, because there are some alloreceptors that are specific for the macrophages. What we're talking about is a more general activation of the myeloid cells. And this was indeed the rationale why some people during the COVID pandemic said, why don't we get before the vaccine was available, why don't we get another sort of vaccine to train our immune system to something that is similar to the COVID, even if it's not exactly the same antigen. So what we did, it's possible to train the macrophages in vitro. So here is when we give LPS, they start producing a little bit of cytokines. But if we expose the macrophages at day 0, okay, with a beta-glucan, and then we expose after 1 week to LPS, they start producing much more TNF-alpha and IL-6. This is what is called the training, because when they are exposed to the same stimulus, they produce much more cytokines. However, when we give at the time of training erythropoietin, or when we give it 3 days after, this is significantly reduced. So EPO can inhibit the training, so the memory of the macrophages. And we actually checked the DNA, and we saw that these epigenetic changes that are induced by the training are inhibited by EPO in the genes that encode for TNF-alpha, IL-6, and other genes. We showed the same also in vivo. If we train the macrophage, the mice, they respond much more to the same stimulus, to LPS, but not if we give EPO. And EPO is not really working if the mice don't express the EPO receptor. So these are, we thought, were quite exciting results. And then we said, okay, if this is the reason, let's try to give EPO, the CpG before the transplant, with the idea that patients come to the transplant with an already acquired trained immunity. That could be for the different infections, for dialysis, multiple reasons. If we give EPO to mice that have already been trained, as you can see here, we can restore the tolerance in mice that receive CTLA-4 And so this is when we gave the EPO at the time of the training. We don't really know when the training is happening. So also when the training happened weeks before the transplant, and we gave EPO at the time of transplant, as you can see here, we could restore the graft acceptance that was broken by the training of the immune system. So EPO has effects on the T cells, on the antigen-presenting cells. We started wondering whether EPO has also some effects on the B cells. And so to do so, we immunized our heart transplant mice. We measured the DSA, the donor-specific antibodies, and we saw that if we gave EPO, we saw a reduction of the antibodies. The antibodies were still there, but there were fewer antibodies. They were still there to a level that the graft was rejected, but there was a reduction. And we saw that there was indeed a reduction of the germinal center B cells, of the cells that are encountered an antigen and saw the help of T follicular helper cells. We saw also a reduction of these T follicular helper cells and an increase of the T regulatory cells. Okay, so these are the equivalent of the T effector and T regs for the B cells. And then we asked the question, do we see less antibodies and less B cells because EPO is acting on the T cells and the B cells, or just the T cells? And when we treated the mice that do not express the receptor on the T cells, we found that so EPO was inhibiting the TFH, but these mice became insensitive to EPO. And interestingly, we saw that in these mice, there was also no change in the amount of germinal center B cells. So indicating that the effect of EPO on the B cells is not a direct effect, but is mediated by the T follicular helper cells. When we removed the EPO receptor on the B cells, we saw no effect of EPO on the B cells. So this is the working model that we have, in which EPO, we think, prolongs the graft survival by inhibiting effector cells directly and through the induction of Tregs, and by inhibiting the help that the B cells are being provided, so they produce fewer antibodies. We are interested in transplant, so most of our studies were done initially in transplant patients, but we started asking whether EPO had also immune-modulating effects in autoimmune diseases. This is a paper we did in mice that are prone to develop lupus. And when we treat them with erythropoietin, we saw lower albuminuria, lower anti-double-stranded DNA antibodies, and less severe disease in the kidney. So indicating that the same mechanism that apply to transplant apply also to autoimmune diseases. And in all our studies, we gave exogenous EPO. So a question that I always receive is whether the endogenous EPO has a role in peripheral tolerance. And these are studies done by others showing that the macrophages that express the EPO receptor, this EPO receptor is sensing the endogenous EPO. And by activating the EPO receptor, macrophages become also more sensitive. They can phagocyte, eat the apoptotic cells, and so allowing the clearance of these cells. If that doesn't happen, these mice progressively develop antibodies against anti-nuclear antibodies, and they start developing lupus nephritis, probably because of the accumulation of these apoptotic bodies, which is one of the mechanisms of lupus nephritis. We did similar experiments. We developed now mice that in which we can silence EPO production. Okay, we have with a silencing RNA. If we give dox food, there's a downregulation of the EPO gene, of the EPO levels, and progressively, months after the downregulation of EPO, also of the hematocrit. And we watched these mice for over 5 months. And what we saw is that progressively, similar to the data that we saw before with the lupus, they started developing anti-double-stranded DNA antibodies. Progressively, they develop a clinical score of lupus severity of the skin lesions and albuminuria. And also in the kidney, we started seeing infiltrates, and these mice died faster. And they didn't die because of anemia. They died because of this systemic autoimmune disease. So EPO, the endogenous EPO, is important to preserve self-tolerance. If we remove EPO, we start developing antibodies against self. We also saw that in the mice that don't have EPO, there's an upregulation of these activated B cells, and there's also fewer of the T follicular regulatory cells. So most of the data have been generated in mice, a few data in pigs. In collaboration with Joren Madsen at MGH, we also tested EPO in non-human primates. And so we started asking also what is the effect of EPO in humans. And this is a recent study in patients with polycystic kidney disease. These patients have a high production of erythropoietin. And after transplant, these patients tend to have higher level of EPO. And it seems that the level of hemoglobin was one of the main predictors of eGFR at one year. This is not proving anything, but is consistent with our hypothesis. In patients with polycythemia vera, these are patients that do not have more EPO, but have a chronic activation of the EPO receptor. Well, also in these patients, there's higher levels of Tregs versus healthy controls. Again, suggesting that EPO receptor signaling is important for Treg formation in humans in vivo. But these are kind of extreme situation. Is the EPO that we use to treat anemia in patients with chronic kidney disease having any effect on the immune system? So this is a study that was done in patients with rheumatoid arthritis that were treated with the same level with the doses that are normally used to treat anemia. And in these patients, erythropoietin was able to increase the well-being of the patients. This could be related to the fact that these patients were no longer anemic. But this, again, is suggestive that there is an immune-modulating effects in human in vivo as well. We tried to test it in a more formal way. These are patients with chronic kidney disease. We analyzed the Treg before and at 6 months after the initiation of erythropoietin because of anemia. And we also had a parallel group of patients that did not receive an erythropoietin. These patients, as you can imagine, had the same level of GFR, but were primarily patients with polycystic kidney disease, and so they did not have anemia. And what we found is that in controls, there's the level of Treg does not change over time. But in patients that receive EPO, there was a significant upregulation. There was twice as many Tregs as at baseline, which could, again, could be due to other reason, but this, again, is suggestive that our model is working also in humans. And these are patients with CKD, now in collaboration with people at the Charité Hospital in Berlin. We are giving erythropoietin to kidney transplant recipients, and we are measuring the Tregs before and after erythropoietin administration. And this is the status of the grant. And this is the study that started, I think, our collaboration with Josh. The idea was to test a single dose of EPO in patients that are otherwise stable. And we decided to study patients with autoimmune hepatitis, which is a condition that is associated with deficit in the number and function of Tregs. And then we took serial samples and we analyzed them with cyto. These are the characteristics of the patients. The patients were stable, on stable immune suppression. The EPO was a single dose, was very well tolerated. All the liver enzymes were fine. And more importantly, there was not a significant increase in hemoglobin. And IgG levels were fine, but the patients were in remission. And what we found is that there was a significant upregulation of the Tregs at 2 weeks that progressively declined. So this is we tested this in a more formal way. So these are stable patients. We gave EPO, the Tregs go up, and then if we wait 3 months, what happens is that the Treg go back down. And the extent of the increase, okay, from 4 to 6%, 50% increase, is what others have shown with IL-2 or other kind of treatments. So it's not a negligible increase with just 1 dose of erythropoietin. And Tregs, as we said initially, is a heterogeneous family in which there's many subsets. And so we did very comprehensive analysis of the Tregs. So we wondered whether EPO was affecting not just the number, but also the composition of the Tregs. And we found that after EPO, the Tregs were not just more, but were a little bit different. And in particular, we saw an increase of these cells that were Tregs expressing HLA-DR, which has been shown to be a marker of increased suppressive activity. And so what we did is we tested the suppressive function of these cells. We took the Tregs before and after EPO, and we added to effector cells the same amount of Tregs, just taken before versus after. And as you can see, the proliferation of the effector cells, if we use the cells taken after EPO, was much lower. So the suppressive effect went up. So the Treg expand, the Treg change in their composition, and they're more suppressed. Okay, so all these data led us to see how can we leverage this knowledge and how can we go to the next level. How can we take advantage of what we know? And we decided to focus on this problem, the fact that the complete immune suppressive withdrawal is feasible in carefully selected liver transplant recipient, but the rate of success is low, is much higher than in kidney, but it's not everybody. And unfortunately, we don't know which ones are the patients that are going to tolerate this maneuver. So the idea that we had with Josh is, can we use EPO to expand Treg, so to allow the withdrawal of immune suppression in a larger fraction of liver transplant recipients? And we said EPO is increasing Treg induction through the production of TGF-beta, is inhibiting effector cells. But based on the experience that Josh had, we could also combine this treatment with everolimus, with mTOR inhibitor, that is also increasing the expansion and the stability of Tregs. So this was our idea: take patients, liver transplant recipients that are stable on tacrolimus, and then switch from tacrolimus to everolimus, start erythropoietin, and then progressively reduce everolimus. This took a few years after the initial idea. And we now have a name, and as you heard, we just received the formal notification of funding, and this is the design of the study that will include stable liver transplant recipients 1 to 10 years after transplant, and they will be switched from tacrolimus to everolimus, and then they will receive five serial doses of erythropoietin. This will be a phase 1b study, safety study, but will give us also hints about the efficacy of this treatment. We will have mechanistic studies, and we will be able to understand how many of these patients at 1 year after the maneuver will be able to tolerate withdrawal, and we will also repeat the biopsy and see if there are infiltrates or not. This is not a randomized study, but we decided to do in such a way that patients will be on tacrolimus, and they will be spend some time on everolimus alone, everolimus plus EPO, or EPO alone. So this will allow to test us to distinguish between the contribution of everolimus, erythropoietin, and the combination of the two within the same patient. Okay, so, and the idea is that, of course, we want to withdraw immune suppression because immune suppression increases the risk of cancer, of infections, but there's also other metabolic effects. So we will check the level of the glucose tolerance and other parameters. But it's also possible that, and this is a study that I just found recently, that EPO has other direct effects that are not erythropoietic, that are not immune-mediated. So these are mice in which EPO increased the hematocrit, but also reduced the body weight and the accumulation of fat. And there's multiple mechanism how EPO is doing so, but it's possible that we will capture also some signaling that is not just related to the fact that we will withdraw immune suppression, but there will some beneficial effects of erythropoietin. So today we started with erythropoietic effects of EPO, and we discussed a lot about the effects of EPO on the immune cells. There's also many other effects of EPO on many other cells in the organisms. And EPO is quite evolutionary conserved. Insects have erythropoietin, and very simple animals have erythropoietin, suggesting that even if the erythropoietic effects is one of the main mechanisms, probably EPO has other mechanism of surveillance of the autoimmune response and health of our vascular cells and fat cells and myofibroblasts that are really important. So this is my last slide. I want to thank all the people in my lab that contributed to this study, including particularly Peter Higer, that was my mentor at Sinai. And I really want to thank Josh that allowed to move this study and translate them into a clinical trial. We also had help from people from the Massachusetts General Hospital and from different funding institutions. Thank you for your attention.